Hello everyone, I'm Neil. Today, I'm going to talk about the genetic testing of molecular diagnosis and sharing some information about what advantage of sensitivity and simplicity that BiOptics can provide in the genetic testing field. And this is the topic. First, I'll briefly introduce about molecular diagnosis, and then I'll introduce the technologies which BiOptic has, and share a few applications we have provided now. And then I'll show you the BiOptic COVID-19 test. I think we're still in the chaos. Many people around the world are still sick, and some recent reports said that the vaccine may not be as effective as expected, and it may not even be resistant to mutant virus. Therefore, isolation and screening are still the most effective way to control the epidemic. I'll show you what solution BiOptic provides. Okay, what is molecular diagnosis? In the past, for diagnosis and analysis, doctors or scientists often analyze samples from a human or organism through pathological method. For example, to observe morphological change and biochemical features in tissue, cell, blood, urine, or other body fluids. With the advancement of molecular biology, many current diagnosis and analysis have been improved. Molecular diagnosis is a collection of techniques used to analyze biomarkers in the proteome and genome, and also how cells express their genes as proteins. We use molecular biological technology to monitor disease, detect risk of infection, and decide which therapies will work best for individual patients. It provides fast and high accuracy results. For the diagnosis of proteasome, the basic principle is based on immunology, and the diagnosis of genomics is to analyze the nucleic acid in the sample. The common quick screening, the type of test in which the result can be known in 5 to 10 minutes, usually belongs to the proteome part, which is the immunological detection of antigens and antibodies. Its principle is actually similar to the pregnancy test, and the product design is also very similar. It is lateral flow assay. An antigen is a molecule that can stimulate an immune response. They can be proteins, polysaccharides, lipids, or nucleic acids. Each antigen has distinct surface features that are recognized by the immune system. An antibody is a Y-shaped protein produced by immune system B cell. The tip of each Y-shaped arm contains antigen binding sites. It can bind to a specific portion of antigen. The advantage of using proteomics for molecular diagnosis are fast, cheap, and easy use. However, usually the sensitivity and accuracy is not so good, but it is still a worldwide used technique in molecular diagnosis. And the other part of molecular diagnosis related to the genome is to analyze the DNA or RNA in the sample. Common method is polymerase chain reaction, PCR, and reverse transcription PCR. PCR is a method widely used to rapidly make millions to billions of copies of specific DNA sample. It allows scientists to take a very small sample of DNA and amplify to a large enough amount to study in detail. PCR was invented in 1983 by American biochemist Harry Mullis. It is fundamental to much of genetic testing, including analysis of agent samples of DNA and the identification of infectious agents. The principle of PCR is to use repeated temperature rises and drops to unwind and open the double-strand DNA and let the primers anneal and then polymerase start to work. And using PCR, copies of very small amounts of DNA sequence are exponentially amplified in a series of cycles. Since PCR technology has been developed for decades, there are also many extended applications and transformations. Here, I will introduce two technologies that are more related to today's topic. One is real-time PCR, and the other one is reverse transcription PCR. The two of them are often confused. Just because the abbreviations are both RT-PCR, let's see what's the difference. At present, the most familiar and popular nucleic acid detection method is real-time PCR. Many people call it qPCR, quantitative PCR, or RT-PCR. But in personal, I think RT-PCR may be confused with reverse transcription PCR, so I always say real-time PCR. Real-time PCR, as the name suggests, measures the amount of PCR product at the end of each amplification cycle. To do this, you label the PCR products during or after the primer extension. There are two principal methods of DNA quantitation in real-time PCR. One uses DNA intercollecting dye or minor groove targeting dyes that show enhanced fluorescence upon binding. With each amplification cycle, once the template DNA replicates, the number of fluorophores bound and the intensity of the fluorescent signal increase. Common fluorescent intercollecting dyes are cyber-green 1 and cyber-gold. Their primary advantage is cost-effective, but they are non-specific and bind all double-strand DNA and not just the target. The second method uses sequence-specific DNA probes, which is fluorophore labeled. A variety of chemists are used for detection. The most popular type uses the exonuclease activity of Taq polymerase to chew up oligonucleotide, which has a fluorophore and a quencher at 5' end and 3' end. The exonuclease activity of the Taq polymerase helps separate the fluorophore from the quencher, which results in increased fluorescence. And this is reverse transcription PCR, which the major target template is RNA. RT-PCR, the RNA template is first converted into complementary DNA, cDNA, by using the reverse transcriptase. The cDNA is then used as a template for exponential amplification by using PCR. And of course, combined with real-time PCR, it can be RT-qPCR. And we simply compare real-time PCR and endpoint PCR. For real-time PCR, it can do quantification, and it provides high accuracy. It is a closed system to reduce risk of contamination and no need for post-analysis. But it still has several limitations. It cannot monitor amplicon size, multiplex target limitation. For now, 4 or 5 targets are allowed, so it also depends on instrument. And usually every lab have a PCR machine, but not every lab have a qPCR machine. And for real-time PCR, the technician needs higher technical level than using regular PCR. For endpoint PCR, the advantage are using cost-effective materials and instruments, and it can do multiple targets as long as the signal peak can be separated and distinguished. And low technical level, I think basically as long as you can use pipette, you should be able to do PCR. And the limitation of endpoint PCR is non-specific amplicons and no quantification. In fact, each of them has its own advantage and limitation. So it still depends on the user's testing situation to make a choice. So, here's the common application of genetic testing. There are numerous applications for PCR. It is commonly used for both diagnostic and basic research in different fields. Diagnostic use. Diagnostic PCR is applied to rapidly detect nucleic acids. For example, infectious disease and gene abnormal which may cause cancer or other disease. And detection of GMO. Some modified gene has their specific sequence. We can use PCR to identify them. Forensic testing. Using genetic testing technology, investigators not only collect fingerprints from crime scenes, but also obtain unique information hidden in each person's DNA. And veterinary molecular diagnostic. Veterinarians utilize PCR technique can detect the very low copy number of microbials and animals' body fluid. It is more sensitive and accurate than using a microscope to see the slide. The global molecular diagnostic market size was estimated at about 36 billion in 2020 and is expected to reach 44 billion in 2021, and the largest part is PCR. As you know, BiOptic is a biotechnology instrumentation company. In the past, the most impressive technique of BiOptic is about capillary electrophoresis, CE. We have worked on CE for over 15 years, and we have dedicated in providing an on-site total solution of detection platform and methodology. So, in recent years, we transferred part of our R&D energy to reagent development. And let me introduce the core technologies of BiOptic genetic testing. In usual procedure of genetic testing, there are 3 steps. First, sample collection, and second, sample extraction, and then add the sample extract into the premix reagent to do PCR. In fact, the most troublesome step is extraction, unless you have a fully automated instrument, and as I know, it is usually very expensive. Otherwise, you must step by step to do extraction, and it takes at least half an hour, and if you have more samples, you will even need more than an hour. Also, the extraction kits are not cheap. It costs about $5 for each sample. So, to simplify this step, we provide the extraction-free lysis buffer. No need for complicated hands-on work. Also, no huge centrifuge. Just mix your sample with lysis buffer. The extraction-free lysis buffer is designed for provide easy room temperature lysis of cells or microorganisms. The buffer is a two-fold working solution that can be mixed directly, even with virus-containing sample. After 10 minutes incubation, viral particles and cells are lysed and are ready for direct react in subsequent PCR or RT-PCR. In addition to lysis buffer, if the composition of your sample is more complicated and there will be some impurities that will affect the subsequent PCR, we have also developed a new tool called Q-dip. There is a membrane in front of the stick. Just stir the membrane in a lysate for a few seconds and then wash the membrane in the wash buffer. And finally release the membrane directly to the PCR reagent to start the PCR. It is a simple operation. And in PCR amplification part, we provide direct go premixed reagent. It is a premixed ready-to-use solution containing reaction buffer, DNTP, glycerol, PCR enhancers, and hot start take DNA polymerase. Comparing with conventional take polymerase, direct go prevents non-specific amplification, and it also poses 2 times faster elongation rate than conventional take. Direct go premix is suitable for PCR amplification, even directly from whole blood and samples from cell lines or tissues. You may skip any private DNA purification steps, only applying a tiny amount of raw material directly into the PCR reaction. And it is stable in 25 degrees, so it is convenient to shipping. And for RNA target amplification, we provide one-step reverse transcription PCR reagent. It is used to directly amplify RNA samples. The reverse transcription and PCR steps are both conducted in the same reaction tube. All required reactants are premixed in it. It reduces the hand-on operation and time, and also the most important is to reduce the contamination risk. After introducing the materials, let's see what BiOptic have in the device part. This is the small PCR machine, Q-Amp Mini. It is palm-sized and about 1 kilogram. And we have pre-programmed chip. Just insert the chip to run the program. Even non-bio background users can operate it. And the pedal heating and cooling module positioned at the center. So the temperature uniformity is better on Q-Amp Mini. We can even connect to the car to charge. This kind of instrument satisfies the possibility of on-site testing. And about the pre-programmed chip, you can connect to your laptop and set the PCR program. Okay, then the main product of BiOptic, capillary electrophoresis QSEP. In the middle is the classic model QSEP 100. It can run 96 samples automatically. And we got the protable model QSEP 1 and QSEP 1 Lite, and the high throughput model which can analyze 4 samples at a time. It provides simple operation and no need for sample pretreatment. Just put on your sample, select the sample position, choose the analyze method, and click Run to start. And below figures are the results of capillary electrophoresis. The program can fully automatically interpret your results and also generate the report file, either Word, PDF, or Excel. But since the topic of today's session is not capillary electrophoresis, if you need more information about QSAP series, you can contact to our sales team. And we have also using the same samples to compare the sensitivity of qPCR and QSAP. For this sample, which is about 3 copies, will result CT42 on qPCR. And for CT over 40, usually interpret as negative patient. But in our system, the peak is still as high as alignment marker. Signal to noise value is about 300. So we can say that the sensitivity of CE is very powerful. You can even use CE as a tool for the second confirmation of qPCR, because sometimes when CT is near the threshold, qPCR needs to be repeated again. But if you have QSAP series, just directly put the qPCR product on CE, then you can confirm the result. Based on the previous technologies, what application BiOptic provides? Here, I will introduce 3 model types: ALDH2 genotyping kit, ApoE genotyping kit, and meat identification kit. ALDH2 deficiency, which is a common symptom of alcohol intolerance. It is caused by a single point mutation, SMP, which causes low enzyme activity of ALDH2 protein. So, if you got mutant ALDH2, acetaldehyde may accumulate in your body and let you feel bad. By the way, acetaldehyde is classified as group 1 carcinogen by WHO. Through the special primer design, we can know whether your ALDH2 is homo wild type or homo mutant type or heterotype. And it only needs one single PCR test, no need for gene sequencing. The workflow is quite simple. Just mix your saliva with lysis buffer and wait for a few minutes reaction. And then add the lysate into the premixed reagent, which contains primers and direct go. And then start the PCR. After PCR finished, we can use CE to analyze. So from splitting saliva to final report, you only need about an hour. And for ApoE genotyping, ApoE is also a SMP issue. It has 2 SMP positions on the gene. Apolipoprotein E is a 34 kilodalton protein involved in fat metabolism. It is located at human chromosome 19 and associated with Alzheimer's disease and cardiovascular disease. It has 6 genotypes. According to previous studies, if there is an E4 in ApoE, may cause high risk of late onset Alzheimer's disease. For ApoE genotyping kit provides a fast and simple method of direct multiplex PCR without DNA extraction in whole blood or saliva. In food safety field, BiOptic provides meat identification kit. The original development opportunity was actually from IKEA meatballs. How do we know the real species of the meat that we are going to eat? In this kit, we utilize direct go PCR reagent with specially designed multiple primer pairs to directly amplify the gene from the sample. BiOptic meat identification kit can distinguish beef, pork, chicken, sheep, and goat. And because of high sensitivity of whole platform, the meat can be successfully tested for severe damaged meat products after cooking and seasoning. We even test McDonald's Big Mac and wanted to see if we could make a huge lottery bonus. And because Processed foods often contain unknown materials that will affect the PCR. The sample preparation here we use Q-dip that I have mentioned earlier. Okay, of course, based on the core technology, BiOptic has also developed a convenient, fast, high accuracy, and high sensitivity reagent kit for COVID-19 nucleic acid detection. The current global epidemic is still severe. It's almost 150 million people have got COVID-19. The real number must be several times than that. So, isolation and screening are still very important. Simple and fast methods are needed to reduce the loading on medical staff. Here, BiOptic provides an extraction-free COVID-19 genetic test. This is our workflow. First, prepare the reagent. Mix primer and enzyme. Second and third, collect your sample and mix with protease cake. Fourth, lysis step. And then add the lysate into the reagent to do RT-PCR. And after RT-PCR, directly analyze by capillary electrophoresis. Whole test takes about an hour and a half, and the operation is as simple as you can. As we know, using nasal swab to get the upper respiratory specimens is painful. People may barely accept 1 or 2 times, but I think no one wants to do it very often. So, at the beginning of kit development, we focused on sample collection in an easy. Now, we allow 2 sample collection methods. One is saliva collection. Just rub your gum and buccal mucosa with your tongue and suck your cheek and pour the saliva in your mouth. Then, use the tiny droppers to transfer the saliva into the tube. The other sample collection method is buccal collection. Use the swab to rub and rotate the brush on buccal mucosa for about 10 times. Also rub and rotate the brush on gum. And then insert the brush into the collection buffer. In our kit, we use lysis buffer to simplify the conventional extraction step. So, just add the same volume of lysis buffer with your sample and wait for 10 minutes incubation. During sample lysis, you can prepare reagents. Just mix primer solution with enzyme solution. And after sample lysis, add 5 microliter lysate into the reagent, and then put it on your PCR machine. And this is the reverse transcription PCR program. After RT-PCR finish, just take out the tube and place it in the capillary electrophoresis. About BiOptic COVID-19 test, we detected 2 genes of SARS-CoV-2. One is envelope region, E gene, and the other one is nucleocapsid region, N gene. And the internal control specific to the human RNSP. This figure is the result of positive control. The pink peak is alignment marker. Alignment marker is used for pattern alignment and are used to calculate the size for each peak. Our Q-analyzer program can also identify the peak. The interpretation is automatically. And this is the interpretation of results. And this is our LOD test. it can achieve 20 copies in a reaction. And according to the recent research, most of the infected people have viral loads greater than 100 copies. For the detection of mutant strains, BiOptic also developed another kit. At present, most of the COVID-19 that is prevalent in the world are mutants, especially the mutants in UK, South Africa, and Brazil. They all have N501 mutant. For this N501 SMP, it will cause spike proteins position 501 asparagine replaced by tyrosine. And these variants are more transmissible than the original strain. As we have experiments in ALDH2 SMP identification, we designed specific primers that can distinguish as N501 or as Y501. And I think we will release this kit in the near future. In addition to the above COVID-19 mutant detection, we also have another branch line working on detecting COVID-19 and influenza A and B in one reaction. For now, we can distinguish COVID-19 and flu A. And next step, we will add flu B's primer to test it. In addition, a lot of people have recently proposed a good concept: surveillance testing, surveillance pooling. Especially large institutions and units are most suitable for such detection methods. For example, a large company can classify by different departments, and the employees in same department can mix the specimens together. Because individual examinations are wasting money and time. As long as one person gets sick, people in the same environment should also be isolated and then test for confirmation. This method is more suitable for routine testing in areas with severe epidemics. The part introduced today is actually mainly the application and templates of some concepts. We also welcome partners who are interested in genetic testing to work together to develop more useful and commercially available products. And last, our capillary electrophoresis not only analyze PCR product, but it also have numerous applications. Basically, as long as the related substance of DNA, RNA, and even protein can be analyzed. We have many different types of cartridges that can be used, depending on what sample you are analyzing and what kind of analysis you want to do. And okay, you can join our website to take a look. Okay, and last, thanks for your listening, and I hope that after the epidemic stabilizes, you can visit Taiwan. Taiwan is beautiful and safe.